this. Okay, everyone, thanks for joining us. And there are some people here in person and who braved the rain. So they get extra, they're going to get lunch, which by the way is a little bit late, but we'll be here. So feel free to get up and get your sustenance to warm you up. First, in terms of we're going to start with our land acknowledgement. We'd like to acknowledge the Ramaytush Ohlone people, who are the traditional custodians of this land. We pay our respects to the Ramaytush Ohlone elders, past, present, and future, who call this place, the land that UCSF sits upon, their home. We are proud to continue their tradition of coming together and growing as a community. We thank the Ramaytush Ohlone community for their stewardship and support, and we look forward to strengthening our ties as we continue our relationship of mutual respect and understanding. Okay, so I'm going to go ahead and introduce our speaker. It's Dr. Stephanie Gaw. She joins us from the UCSF Department of Obstetrics, Gynecology, and Reproductive Sciences. She's a leading expert in maternal-fetal immunology and placental biology and has dedicated over 20 years to studying malaria, both in the field and in the lab. Dr. Gaw advises organizations like the NIH, WHO, and CDC on global health challenges and has had research on COVID-19 in pregnancy, Zika virus in Brazil, and malaria in Uganda. Today, she will share her expertise on placental malaria and its significant impact on maternal-fetal health and birth outcomes. Thanks for joining us, Steph.
Great, thank you, Roberta, for that introduction. Let's see. Okay. So thanks so much for the invitation to speak to my pediatric colleagues today. Very much enjoy working with many of you. And I think that a lot of my work really reflects how much OB and pediatrics really integrates in improving health globally. I have no disclosures. So malaria in pregnancy affects about 125 million pregnancies per year, mostly in sub-Saharan Africa. The most virulent strain is the falciparum species, which causes placental malaria. It's estimated that 50 to 85 million pregnancies are actually affected globally by falciparum malaria. Usually in pregnancy, it's asymptomatic. And so diagnosis does not happen, if at all, until after delivery. Primogravidas or first-time moms are affected significantly more than multigravidas. And this is a really interesting phenomenon that is really highlighted in malaria work, but also has implications for other perinatal complications as well. And vertical transmission is rare from the mother to the fetus. And while we think of it as a tropical disease and global, we do know that with climate change and global warming and more global migration patterns, that the border or the barriers to the geographic barriers to a lot of these tropical diseases are expanding. In 2023, there were 7 cases of locally acquired malaria in Florida, Texas, and Maryland. Thankfully, none reported in 2024. But I do think this is something that we, over the next 10 to 20 years, the geographic distribution will be changing. So Plasmodium falciparum. So I think most people know that it starts with the bite. You get it from a bite of an infected mosquito. Let's see. The mosquito injects sporozoites into the bloodstream. There's a short liver phase initially where the sporozoites replicate, and then the liver stage ends, and the merozoites are released from the liver cells, and this goes to establish the blood stage, the asexual blood stage infection. So that's depicted on the left. You can see my marker. Anyways, and the asexual blood stage is responsible for all the clinical manifestations of malaria. So this is where you get the typical cyclical fevers every 48 hours. The infected red blood cells do travel to other, all the different organs. And the most significant consequences are cerebral malaria, which primarily affects young children, where infected red blood cells are sequestered within the brain vasculature. It can cause seizures, coma, and significant brain damage. And then also for pregnancy, these infected red blood cells can sequester within the placental intervillous spaces. And this leads to placental malaria, where we see sequestration of infected red blood cells within the placenta. So I wanted to talk briefly about hemozoin, which is called malaria pigment. And this is, hemozoin is formed when heme detoxification occurs within the blood stage parasites. And as the parasite grows within the red blood cell, it digests hemoglobin and uses its amino acids and other peptides, and it converts heme, which is toxic, to an insoluble and bioinorganic crystal called hemozoin. And natural hemozoin is coded in parasite DNA, and so this can actually stimulate TLR9 responses and is pro-inflammatory. And so hemozoin, while it's a transient byproduct of malaria growth within the red blood cell, it's actually a really important marker of placental malaria, in that even after the parasite is cleared with treatment or with natural immunity, we do see the remnants of hemozoin within the placenta as evidence of prior infection of the placenta. So again, in placental malaria, we have sequestration of infected red blood cells. And here you see a normal placenta on the left, and you can see the really nice placental villi outlined in the red dots. The intervillous space is relatively clear with some red blood cells. And then on the right side, you see a placental malaria slide. And here you see kind of infiltration, more kind of clogging of the placental intervillous spaces, which is the space of the maternal blood. The arrows and the little black dots denote the parasites growing within the red blood cells. This is with CD68 staining, so staining for a monocyte macrophage marker. You can see there's infiltration of maternal monocytes within the intervillous space as well. The placental villi are damaged, essentially, that there's areas of denudation noted by that arrowhead on the upper right. And then SK stands for syncytial knot, where you can see kind of clumping of the normally like the nuclear monolayer of the syncytium that lines the placental villi. Placental malaria has been associated with adverse obstetric outcomes, and in a meta-analysis of multiple studies, it increases risk of low birth weight, which is a birth weight of less than 2,500 grams, preterm birth, small for gestational age, where the baby weighs less than 10th percentile for less than 10th percentile for gestational age, and then also maternal anemia, which is a significant cause of maternal morbidity. And so some of our early studies showed that the syncytial trophoblasts, or the syncytial lining of the placenta, and CD68 positive cells within the placenta actually engulf hemozoin, the malaria pigment byproduct. And again, studies have shown that hemozoin does stimulate pro-inflammatory cytokine and chemokine secretion. This has been shown in monocyte macrophage models and in vitro endothelial cells, and also syncytial trophoblast primary cell lines and trophoblast cell lines as well. And so one of the things that my group has been studying for many years is how malaria infection of the placenta impacts both the maternal and the fetal macrophages that are both kind of simultaneously present within the placental spaces, right? So again, staining with CD68, which is a very reliable pan-monocyte macrophage marker in the placental biopsies, we can see that there are macrophages in the villus side, which is traditionally considered fetal origin, and then the CD68 positive cells in the intervillus spaces are considered maternal. There are, although our definitions of immune cells have changed over the years and essentially gotten much more complicated for all cell types. But I think the general paradigm still is to think about M1 and M2 type macrophages. although there's a huge spectrum in between. M1 type macrophages shown on the left is more on the Th1 pathway. These are thought to be more pro-inflammatory. They're activated. They exhibit phagocytic activity. I think this is what you generally think of when you think of a macrophage responding to an invasive pathogen to try to engulf it and kill it. The M2 type, or the Th2 pathway, is more of and it's called the alternatively activated pathway. Immunomodulatory macrophages, anti-inflammatory, tumor-associated macrophages. And on this side of the spectrum, the macrophages are more, I think, wound healing, calming, and try to temper the immune activation, the inflammatory response, usually we think of with resolution of infection. CD163 is a very reliable marker for M2 type macrophages, so we'll use this in a lot of our studies. So one of the first things we did was look at how just kind of the numbers of maternal versus fetal macrophages in response to placental malaria. So this is using a cohort of placental samples that we had gotten from the Democratic Republic of Congo many years ago. All of these patients were symptomatic at the time of delivery with fevers and positive blood smears. Very limited prenatal care, if any, that we know of. And what you can see here is that when we look at on the very far right of each graph, basically all placentas, right, primigravidas and multigravidas, that in the setting of placental malaria in the dark gray, there is an increase in macrophage cells, basically, both on the fetal and the maternal side. When we look at the activation state of these markers, so we measured this by looking at the percent of the percentage of macrophages that co-express both CD68 and CD163, the M2 marker. And we found that on the fetal side, that an increase in the percentage of the M2 type fetal macrophages or Hofbauer cells correlates with increased birth weights, but really just in primigravidas, not in multigravidas. You can see from the on the left scatter plot. And so this is really interesting. This is really interesting, kind of one of the first times we had seen differential responses to placental malaria based on gravidity. And the association with the clinical outcome of birth weight. We then looked at type 1 interferon genes and type 2. So again, kind of along the M1, M2 paradigm of macrophage activation and pro versus anti-inflammatory responses. Type 1 interferons are generally, again, thought to be anti-inflammatory or immune modulatory. Type 2 interferons are, again, more pro-inflammatory. Interferon gamma is the classic type 2 interferon. And again, when we look at this is qPCR, and we look at interferon beta, MX1, and IL-10 transcription levels with placental malaria based on gravidity, we see that overall in multigravidas, some moms that have had more than one child, the levels of interferons are significantly higher compared to their primigravida counterparts. We saw similar trends for some of the type 2 interferons, but not statistically significant. And then when we look at the correlations between interferon beta transcript levels and birth weight, we again saw that the absolute levels of interferon beta transcript correlates with birth weight. So for each log 2 increase in interferon beta, the birth weight increases in the whole cohort by about 143 grams, which I was kind of surprised by. When we break this up by gravidity, this effect really is largely driven by in the primigravidas. So in the primigravidas, first-time moms, the higher your interferon beta levels are in the placenta, your birth weight increased, their baby's birth weight increased by over 300 grams, which we were pretty surprised at how significant the correlation was. When we looked at malaria versus controls, again, this effect is really primarily seen in placental malaria cases, where again, for every log 2 increase in interferon beta levels, the birth weight increased by about 200 grams. So all of this kind of piqued our interest in understanding how the maternal and fetal macrophages in the placenta directly interface and talk to each other in response to singular insult, right? The placental malaria. So you can see that I think what our data shows so far is that on the fetal side, so this is in the intravillous space, or sorry, in the villus core, we have the fetal macrophages that are primarily M1 or pro-inflammatory and secrete interferon gamma. And this is associated with lower birth weights or possibly fetal growth restriction. And then on the maternal side, the maternal monocytes in the intravillous spaces exhibited more of an M2 phenotype that are more anti-inflammatory, higher interferon beta levels, and this correlated with increased birth weight. And so we kind of wanted to look at this further. So again, this initial cohort was with all patients that had symptomatic active malaria at the time of infection or at the time of birth, right, at the time of delivery. So they all had active parasitemia in the placenta, but again, probably the vast majority of patients have asymptomatic malaria during pregnancy, or they're cleared, because in malaria-endemic areas, the WHO recommendation is for intermittent preventative treatment. So every month, moms are presumptively treated for malaria. So a lot of it does end up getting cleared. And so their classification of placental malaria is based on whether there are infected red blood cells with live parasites seen in the placenta. So that's considered active placental malaria versus past placental malaria. And again, this is when we see just the hemozoan crystals in the placenta. And this indicates that at some point there were live parasites there, but are no longer there, at least not at the time of delivery. So what we wanted to know was, what are these monocyte macrophage phenotypes in active versus past malaria, right? Is there something different if the parasites are in the placenta at the time of sampling? And so we were able to look at the macrophage numbers and also the macrophage phenotypes of the placental malaria in a cohort of biopsies that had either active, past, or were healthy controls. So these samples were all collected from Uganda, and the controls had no evidence of malaria at any time during peripheral sampling throughout pregnancy, and no evidence of malaria in the placenta at the time of delivery. And by multiplexing with these 4 different markers, we can distinguish the origin, the patient origin of the macrophages, whether they're maternal or fetal, and then also their phenotype, whether they're M1 pro-inflammatory or the M2 anti-inflammatory type. So CD68, again, in the pink, is the pan macrophage marker that we use for all, to identify the monocyte macrophages. CD163, again, is an M2 marker. FOLR2, the folate receptor 2, was recently reported by a UK group to be a marker for fetal macrophages. And this is expressed on the cell surface as well. And then cytokeratin 7 is a placental cell marker to distinguish the placenta. And so based on these cell markers and the location of the cells in the intravillous spaces, we can identify, one, if they're maternal or fetal, what their activation state is, and then also see whether or not the maternal or the fetal macrophages can kind of transmigrate into the opposing compartments. And so when we look first in the intervillous spaces, we do see that in the intervillous space, the maternal macrophages, both M1, M2, and M2s, are increased in active malaria compared to healthy and past. This you can see in the top two panels. And then on the fetal side, there was no significant difference in fetal M1 density in the intervillous spaces, but there was an increase in a fourfold increase in fetal M2 macrophages within the intervillous space. And this was really interesting because this actually shows for the first time that fetal macrophages do actually transmigrate out of the placental villi into the intervillous spaces in response to malaria. When we look in the villus tissue, so this is in the placental villi, we see that the maternal M1s are significantly increased in past infection. This is not the best graph too. It's a top left graph. The scales are all even so you can compare, but that was significantly increased with past but not active placental malaria, and no differences for any of the other cell types. So again, this shows that the maternal M1, so these are folate receptor negative cells, transmigrate into the placental villi and suggest that the maternal inflammatory response, pro-inflammatory M1 response, may be responding to some villus, some stimulus within the placenta villus, possibly hemozoin. And again, when you look at the macrophage phenotypes and their correlations with birth weight, we see that on the maternal M1 side in the intervillous spaces, it's negatively correlated with birth weight. So there are lower levels of pro-inflammatory maternal cells with higher birth weights. And then the same thing with the M2 cells in the villus spaces. So this is again maternal M2 type macrophages that are transmigrating into the placental villi. So in summary, what we found from these experiments was that in active placental malaria compared to past, the maternal M1, M2 macrophages from the peripheral blood are recruited into the intervillous spaces. The fetal anti-inflammatory M2 Hoffauer cells, or HBCs, transmigrate into the intervillous space, again in the setting of active placental malaria where there are parasites. And in past, where the parasitemia has been cleared, there is an increase in the maternal M1 macrophages that transmigrate from the intervillous spaces into the placental villi. And that this differential activation of the maternal macrophages are associated with lower birth weights. So next, we wanted to kind of expand our investigation into what exactly is happening at the transcriptional level with these macrophages. So what we wanted to do was to do transcriptional profiling of the maternal and the fetal macrophages to understand what genes are differentially activated in placental malaria. Are there differences between fetal versus maternal macrophages? And are there differences based on the gravidity? And the way we did this was we wanted to purify out the macrophages in the maternal space, of which the bulk, over 90%, are maternal, and the macrophages that are located within the placental villi, of which the bulk are fetal. And we went to Uganda to do this. We collected 40 placentas right after delivery, and basically through this sequential protocol, isolated out the maternal macrophages, and then through a series of digestions and purification steps, ended up with the Hoffauer cells, or the fetal HPCs at the bottom. These are sent for bulk RNA sequencing to understand, again, what happens at the transcriptional level for fetal versus maternal macrophages. So, of course, we're very interested in malaria, but first, what happens in normal pregnancy? No one had actually looked at maternal versus fetal differences in normal pregnancy, normal placentas. So, we first did our analysis of 3 primogravidas and 3 multigravidas. And these were patients with no evidence of malaria at any time during the pregnancy through peripheral sampling, and no evidence of placental malaria at the time of delivery. And looked at these 2 different cell types. So the MIMS are the maternal antiphilous monocytes, and the HBCs are the fetal. And you can see that the 2 different cell types are very different. They segregate really cleanly, which is to be expected since they are from 2 different patients. And when we compare transcriptional differences between the 2 cell types, we see that they're, again, very different. They're almost 1,000 differentially expressed genes. When you compare MIMS versus HBCs in normal pregnancies, there's a greater number of differentially expressed genes on the maternal side compared to the fetal side. And when we did pathway analysis, this bar chart shows you kind of the number of genes upregulated in either the MIMS designated in green on the left versus the HBCs in purple on the right. And you could see that generally there was an increase in inflammatory response pathways, migration, adhesion in the fetal HBCs compared to the MIMS. And in MIMS versus HBCs, there was an increase in induction of pathways related to cell organization and differentiation. And then we looked more specifically at macrophage activation markers. So our prior study that I showed you, really we're looking at, honestly, one activation marker, right, CD163, since we were looking at staining. But there are many different activation markers that are associated with either M1 type phenotypes or M2s. And so we wanted to look at the expression of these M1 and M2 markers in both fetal and maternal macrophages. And I think prior to this work, and I think still most people think that consider Hoffbauer cells or fetal macrophages to be almost all M2 type, and the majority are, but they actually are quite phenotypically diverse. So you can see the HPCs are in purple, and they do express to a greater degree than the maternal monocytes, all the markers to some degree, and many of the M1 pro-inflammatory markers as well. And so I think this really just points out that we have a very, very basic understanding at this point of all the different macrophage subtypes that are circulating in the placenta and what their individual functions may be in not only in response to infection, but in the setting of normal pregnancies. And then when we look at gravidity, we did an interaction with looking at MIMS, the maternal monocytes, and the fetal Hoffbauer cells separately based on maternal gravid status. And we see that only 5 genes are commonly altered by gravidity in both MIMS and HBCs. And so otherwise, the regulation of these 2 cell types is really different based on maternal gravidity, which I think is to be expected. And then a little bit of a preview, we're still working through this data, but in the setting of placental malaria, what happens? So I've shown you that maternal and fetal macrophages are very different, right? Transcriptionally and as far as activation markers go. What about in the setting of placental malaria? Now, all of these cases were cases of past placental malaria. There were no cases of active placental malaria in these samples. And we can see that there were only 19 commonly differentially expressed genes between the two cell types, right? But so the responses of fetal versus maternal macrophages are very different to placental malaria. There were 339 genes that were differentially expressed only in HPCs, and 228 genes that were differentially expressed only in the MIMS. And again, when we look at macrophage markers, we found that most of the macrophage markers were actually downregulated in the setting of placental malaria, that there was very little activation in the macrophages, which implies some sort of kind of tempering of the placental inflammatory response with past placental malaria. And then when we add gravidity to this, what do we see? We see that of the genes that are differentially regulated in response to placental malaria in MIMS and HPCs, 276 were commonly differentially affected by gravidity as well, right? And when we did k-means clustering, we found two main clusters. The first ones were the group of genes that were increased in prima gravidas and decreased in multigravidas in both cell types. And these were associated with the inflammatory response cytokines and leukocyte activation. And in the second cluster, these genes were downregulated in prima gravidas, upregulated in multigravidas, again, in both cell types. And these were related to more cellular biosynthetic processes. So I'm going to switch gears a little bit and then maybe tie it all together at the end. So I've talked a lot so far about placental malaria. What are these macrophages doing? How are they responding? What's going on, in the setting of active placental malaria, where there's parasites in the placenta at the time of delivery, and then also in at the time of delivery, if there is hemozoin in the placenta, there's past placental malaria. Kind of going back to the outcomes, we've talked a lot about birth weight and associations with anti-inflammatory responses and as being protective against lower birth weight. We want to think more about growth restriction and more long-term outcomes. I think in the short-term outcomes, so the initial birth outcomes, we do know that fetal growth restriction, there's a 5-fold increased risk of stillbirth, right? So this is a lot of our prenatal care in the United States is preventing stillbirth. So if babies are growth restricted, we follow them very closely throughout pregnancy. We deliver early if we need to avoid the stillbirth. Even after birth, there's a 2 to 5-fold increased risk of perinatal death. And of course, this depends a lot on gestational age at delivery and overall size. And then a 3-fold increase of other adverse perinatal outcomes at the time of delivery. But there is a whole breadth of literature on fetal growth restriction or small for gestational age babies and longer term outcomes. Fetal growth restriction in developed countries has been shown to be associated with neurodevelopmental delays and abnormal neurodevelopmental testing later in life. There is a really strong association with fetal growth restriction in utero and metabolic syndrome later in life. There is an increased risk of obesity, diabetes, and other metabolic disorders in childhood and adulthood, cardiovascular disease, renal disease, and pulmonary disease as well. So this all kind of ties into the developmental origins of health and disease, or the Dohad hypothesis or the Barker hypothesis, where a lot of adult or later onset medical conditions, the groundwork for that or the predisposition is really laid by in utero environment and exposures. And we think a lot of these are epigenetically mediated because they do have long lasting and in some cases have been shown to have intergenerational effects on longer term health outcomes. So what we wanted to look at was genetic differences in the placenta with growth restricted babies, either from the US where there's no malaria at all, so we coined that non-malarial growth restriction, versus growth restriction that's associated with placental malaria. And these are from samples collected in Uganda. So we designed this pilot study to compare the placental methylation essentially between a cohort of FGR babies born in San Francisco and their respective controls, and then also a cohort in Uganda where the babies were severely growth restricted and also had placental malaria. And this was done with bulk placental tissue. We extracted total DNA and did applied the DNA to a methylation array. And so this methylation array, because we will look at the methylation status of over 860,000 CPG sites. And so we were actually really surprised by our results. So the first thing is when you look at differentially methylated CPGs, so this is after kind of all the data cleaning, we looked at in panels A and B, we looked at the two cohorts independently. So we know a lot of things can affect epigenetics, right? Like diet, environmental factors, genetics. And so looking at in A, it's the non-malarial FGR against their respective controls from the United States. There were only 65 CPG sites that were differentially methylated between the two groups. In panel B below, when we look at malarial FGR, so again, these are samples from Uganda, we saw that there are only 133 differentially methylated CPG sites. Then we thought, oh, there must be a lot of differences based on just location, right? Different patient populations. One population's from Africa, the other one's from the US. There must be differences. But when we look at the control placentas from each of these two groups, again, very few differentially methylated CPG sites, only 183 were different between the two patient populations. And the most interesting thing actually is that the greatest differences were when we looked at compared the two different two different FGR populations. So the non-malarial FGR compared to the placental malaria FGR, there were over 500 different methylated genes. And this really implied a lot of like very distinct epigenetic impacts of FGR specifically and the placental methylation profiles. When we subtracted out the differentially methylated genes from each site, so from the US versus from Uganda, and looked at what CPGs were commonly differentially methylated, there was actually only one. And this is a pretty strong association that this corresponds to a promoter, the CPG site corresponds to a promoter region for a BMP4, bone morphogenic protein. This is hypomethylated in both non-malaria and malarial FGR. And this is a cell growth related gene. It's implicated in trophoblast differentiation within the placenta and has also been implicated in other studies of fetal growth and SGA in other countries as well. But when we look at what is differentially methylated in between the two different FGR types, we actually found that with placental malaria associated FGR, there were 100 CPGs that were hypermethylated, so were silenced, and that many of these genes were associated with pathways related to development and nervous system development compared to non-malarial FGR. And this is really interesting to us because in kind of a parallel study, another cohort of this is a cohort of infants born out of one of our prior IPT treatment, malaria treatment and pregnancy studies in Uganda, where about 200 of the infants were followed up to 36 months of age, and they were very comprehensively tested for many different neurocognitive outcomes. And this study was published a couple of years ago by Paul Bangarana, who is one of the leading neurodevelopmental psychologists in Uganda and in Africa, I think. And what he found was that in this cohort of, about 250 infants, that malaria and pregnancy was associated with reduced or lower Bailey scores at 2 years of age that persisted to 3 years of age, from 24 to 36 months. And this was significantly associated with any type of malaria in pregnancy, and especially more so with malaria pregnancy earlier in gestation rather than later. So malaria pregnancy prior to 20 weeks rather than after 28 weeks. There was a trend towards a lower scores with placental malaria, but this is not statistically significant. And so we actually have been continuing this work in our most recent cohort of malaria treatment in pregnancy. There are about 900 infants born to moms that were treated, randomized to different arms of malaria treatment in pregnancy, and we are following these infants till up to 4 years of age. And so this study is ongoing right now. I think all of the kids will turn 2 by May. And so we've tested so far about 500 of a planned 750 children at the 24-month time point, and then we're going to follow up at 42 months as well. And so I think this larger sample size, it'll be 3 times as much as this prior study, I think will hopefully give us enough power to try and dissect the impact of malaria in pregnancy, placental malaria, plus or minus growth restriction as an independent modulator of adverse outcomes. So in summary, I've kind of taken you through a lot of stuff about placental macrophages and growth restriction and malaria. I've shown you that maternal and fetal macrophages both respond to infected red blood cells and hemozoin deposits, and have different phenotypes as well. We've shown for the first time that maternal and fetal macrophages actually can transmigrate across the villus barrier in response to both past and active placental malaria, and that differential activation of the macrophages and interferon pathways are associated with fetal growth, and that the impact of these pathways is modulated by maternal gravidity, and is different but on the fetal versus the maternal side. The maternal neurovillus monocytes and the fetal Hofbauer cells have distinct gene signatures, and these are also modulated by gravidity and show distinct responses to placental malaria, again in with respect to maternal gravidity as well. And that our newer work, that malaria-associated growth restriction has distinct methylation profiles that's very different from non-malarial FGR that we would see here in the United States. And this has potential implications for longer-term outcomes related to neurodevelopment in offspring that were born to moms that had malaria during pregnancy. So I think that's it. Thank you so much. Obviously, this is a lot of work, and many people have been involved over the years, but the majority of the lab-based work was done by Nito Zarslan, a postdoctoral fellow at my lab. And I'm happy to take any questions.
So my question is related to I have a question about the neurodevelopmental. Maybe somebody else here has thoughts about that. But just to start out, the fetal growth restriction. So I guess my question is around gestational age, because I understand like gestational age accuracy is complicated. So you need
Yeah.
additional age, right, to like determine how restricted they are and their patterns you see across gestation.
Great questions. I'll repeat the question. So Roberta asked how gestational age is determined and how does that play out with the diagnosis of growth restriction. So in the first cohort I showed you, those were patients that delivered with limited prenatal care and all we had was birth weight, right? So we couldn't really assess fetal growth restriction necessarily, but they were mostly born at term, reported term. In all the follow-up studies, these were in the setting of trials. So all of these patients were gestationally dated with OB ultrasound, and they were all enrolled prior to 20 weeks. So I think we have really good gestational dating for the subsequent kind of more molecular studies that we've been doing with our more recent cohorts.
Like I guess, but I understood, and maybe I understood this wrong, but that the women are kind of intermittently being treated. So some of the past malaria could be because they were treated or because they just controlled.
We're actually planning to look into that right now. I think that in this area of Uganda where we conduct these studies, the endemicity of malaria is so high. So about 50% of women enrolled in the studies have parasitemia detected in their blood at the time, and they're completely asymptomatic. So about half of them are treated for malaria upon enrollment because we screen all of them. They're being screened monthly. And of course, they're treated for any, they're getting the standard treatment, right, to prevent malaria or treat malaria during pregnancy. But, if they have any fevers or they're followed so closely, they're treated if they have any episodes during pregnancy as well. At the end of the study, so the study we just completed was about 2,757 women total that were randomized into 3 arms. So a huge study. Only about 200 of them had no evidence of malaria at all at enrollment and through all the monitoring. And so the impact of malaria is really high. When we look at placental malaria based on histology, so again, looking for hemozoan deposits or malaria at the time of placenta biopsy at the time of delivery, again, over 40%, 50% have evidence of placental malaria. So it is pretty, it's highly prevalent, right? And so I think this is where it's really interesting to me to think about the impact that malaria has on the whole population in these really highly endemic areas. It's almost impossible to avoid, right? And these are patients in a trial setting when they're getting basically DOT, like we're watching them take their meds every month. They come in. If they don't show up, we go to their houses. And so even in kind of the optimum conditions for malaria treatment and prevention, as far as we have in pregnancy right now, there's still a 40% risk of placental malaria.
There's a question. Considering using the transcription profiles for drug.
Oh, that's a great question, Jim. I have not thought about that. Yeah. Yeah. So the question is why prima gravidas have a different response to multigravidas. And so I think in the malaria world, it's long been known that the highest risk moms are the first time moms. And I would say probably until very recently, most of this has been chalked up to antibody mediated partial immunity to placental malaria specifically. So there are specific parasite antigens that allow for the sequestration of the parasite in the placental spaces. So there's these kind of placental malaria specific antigens that once you've been pregnant before, you do have these antibodies against, they're called VAR2CSAs. But I don't think that explains everything, right? I think if that were the case, then if there were circulating antibodies, you would have no placental malaria in second or third time moms if antibodies were present, right? And I think, with the work that we've been doing, looking at the innate immune responses, right? The macrophage responses specifically. Even in the normal placentas, there are gravidity-based differences that persist even on the fetal side. So you can see like the mom, of course, the mom's like, different, right? Like pregnancy changes things, and they've also been exposed to other things in between pregnancies. But even on the fetal side, their baseline transcriptional profiles are different. So I think that there is something about pregnancy that can train your innate immune system, right? And especially even early in pregnancy, the fetal macrophages are derived from the placenta, which is or from the early yolk sac, right? Early in pregnancy, and the development of that is really dependent on maternal uterine factors. The uterine macrophages are really important for early pregnancy establishment and development. And so I think that actually pregnancy or reproductive history can train your immunity so that when it does see, some sort of challenge during pregnancy or even the subsequent pregnancy, the next pregnancy itself, that the responses will be different.
The developmental testing, it looked to me like the control scores are actually going down.
The scores are going down over time. And I think, relative. And I think it is, I didn't show it here, but the biggest driver of lower scores is malaria in the first year of life for that infant. Which to me also relates back to malaria and pregnancy also. Yeah, because if you have malaria, if your mom had malaria during pregnancy, that infant is actually at higher risk of getting malaria in infancy. And I think there's some sort of immune tolerance that happens that makes them more susceptible to developing malaria in infancy. That's why I think we're really excited about this study that, is ongoing because, we were able to see these, Paul and his group were able to see these results with, a third of the patient samples or, sample size in the prior studies. So we're hoping to be able to dissect kind of these more complex relationships with the larger sample size. Thank you.
Right.